Okay, want to thank the organizers for this opportunity. So what I thought I'll try to do in my 12 minutes is to give you some broad strokes on where we've gone with inflammatory bowel disease. So I think that Eric had sort of a slide that said genetics started in the mid-'80s. And if you go back to early mid-'80s, this is what we knew about inflammatory bowel disease. There was clearly a genetic association based on family history. And there were some environmental factors that contributed to this disease, and you ended up with inflammatory bowel disease. So I've had the unique opportunity of sort of collaborating with Mark since the mid-2000s, where we've been able to sort of take inflammatory bowel disease genetics from common GWAS all the way to fine mapping, and I'm going to give you a few examples of how we've translated this to biology. What we'd all like to get is this answer. So taking knowledge from human biology to try to test hypotheses that would influence disease positively and negatively, and find features of chemical biology that would potentially correct the disease phenotype. Can we do this? The answer is yes. So in one example where we've looked at IL-10 signaling in inflammatory bowel disease, so if you go back to the 2,200 odd loci, you have about 4 to 5 genes that impact inflammatory bowel disease that affect IL-10 signaling. At the Broad, we set up an assay to look at small molecules that enhance IL-10 in the gut and suppress IL-12 and TNF-alpha in the gut. And that led to the discovery that the SIK family of kinases did that switch. We created a disease-resistant allele so that we could get to specificity, and have now gone on to identify small molecules that induce IL-10 in the gut and suppress pro-inflammatory cytokines. And for us, the good news is the pharma has picked this up. So moving from genetics to biology to small molecules. So the first question that I'd like to answer, is the cell type important? And I'm going to say yes. So with Aviv Regev, we've gone about mapping the expression of disease-associated variants, starting with the simple idea that if you take patients with ulcerative colitis and look at gene expression changes in the unaffected colon compared to the affected colon, one might be able to identify cell types and then pinpoint to genes that might be expressed in those cell types. Using this 3 millimeter biopsy, we can get to something like 50 different cell types from the gut, from the inflamed versus non-inflamed, and then ask questions, where are these disease-associated genes expressed? An example, so a gene called HGFAC, which is a hepatocyte-expressed gene. So at first impression, when you look at that gene, it's named as a hepatocyte-specific gene. But what we identified was that in both in the small bowel and the colon was that this gene was highly expressed in goblet cells. So so-called liver-specific gene that has a high expression in the gut. A second example is if you look at other genes, you can see that there is clearly a pattern of expression across those 50 different cell types. And you can start to ask questions. If you take the expression of gene modules across all cell types for this particular gene C1off106, you end up with sort of a generic signature saying this is an epithelial cell-specific signature. But once we found out that C1off106 was expressed specifically in enterocytes, now we could get to function that this gene regulated cell-cell junctions. And we were able to confirm that earlier this year. So cell type matters and gene expression helps, but often multiple cell types interact to give you disease. And this is best illustrated with autophagy as an example. So autophagy is a process that takes intracellular organelles and clears it to degradation through many cellular processes. And what we and others have identified is that there are at least 3 different processes that autophagy impacts in the gut. And again, with Mark Daly in 2007, we were able to identify that ATG16L1 T300A was associated with inflammatory bowel disease. And here's an example where many cell types express that variant and have many phenotypes, all having the same functional impact. So the disease variant creates a caspase cleavage susceptible site, making the protein more unstable, but the phenotypes extend from Paneth cells to wound healing to production of cytokines to function of regulatory T cells. So an example of where a disease allele now has effects on many cell types contributing to disease in that particular tissue. Can you translate this into some sort of predictive value? And an example here, if you look at the expression and Paneth cells in patients who carry the allele versus the wild-type allele, you can see the time to surgery was much shorter in patients who carried the risk allele. So an example of where you have application of genetics to human disease. When we started autophagy, there was 1 or 2 genes that were associated with risk of autophagy. The next experiment we did was we took all the genes within the 200 plus loci and asked, are there additional autophagy genes that could be present in the loci that have not, up to that point, been implicated in autophagy? Without going into a lot of detail, we've gone on to identify something like 20 additional genes within the IBD risk loci that are associated with autophagy, now making a strong case that autophagy is an important process in mucosal immunity. Just to give you a quick example of what those cells look like, so if you take GPR65 knockout cells, you get these bags of ineffective phagosomal structures and lysosomes that are markedly abnormal. If you look at the risk variant, you have a milder phenotype of these same observations. And as I showed you in my second slide, the goal is to get to therapeutics. And again, we've started to screen for small molecules that can correct the risk allele and recover the phenotype back to the wild-type allele. And we've been able to do that with a molecule that we've identified that can correct the defective variant. So I've given you 2 examples. Identifying a gene, a specific cell type, you can get to function. I've taken a process like autophagy and said autophagy affects many cell types, and to cause disease in a tissue, many cell types have to interact for disease. A third notion that's important is in a disease like inflammatory bowel disease, the notion was that you always had to have lots of inflammation, hyperinflammation to cause disease. And when we started working up one of the risk alleles, we identified that immunodeficiency, so loss of immunity, also can increase the risk of autoimmune disease. And an example here is with this locus. So SP140 is a BTB domain containing transcription factor that's associated with Crohn's disease. I'm not going to spend a lot of time. The SNP leads to loss and truncation of the SP140 protein. And you can begin to see that how this allele might be associated with disease. And if you go directly to patient samples, you can see that it's associated with hyporesponsiveness. So less cytokines, but yet inflammation in the gut. So again, a third example of a disease allele telling you about function and loss of inflammation, increased autoimmune disease, but with low inflammation. The fourth concept that I'd like to highlight is the value of rare variants. So in a gene called COD9, again, we and others have gone on to identify a common allele that's associated with risk, but in that same gene, a protective allele that protects you from disease. Again, we've gone through the process of moving from genetics to biology to mechanism and a therapeutic hypothesis, being able to show that the COD9 risk allele binds a E3 ligase called TRIM62 that fails to bind the protective allele, and that interaction ends up in ubiquitinating COD9 on a specific residue, 125, and that ubiquitination stabilizes the protein, increasing inflammation. The attractiveness of this is that evolution has validated that this COD9 protective allele is a safe and effective strategy for decreasing the risk of developing inflammatory bowel disease, and we've used this experimental system now to identify small molecules that mimic the protective allele. So to summarize, 200 plus loci. The question is, are you going to keep chasing all the pathways in human biology? The answer is no. So if you look at inflammatory bowel disease, they broadly fall into a small number of pathways that are linked to sub-pathways. And again, with Mark, we've been able to systematically map many alleles or disease-associated loci that impact those sub-pathways that contribute to barrier function, inability to handle intracellular microbes, altered production of cytokines, features of defective pathogen sensing, and features in adaptive immunity. So what's the path forward? So what I've shown you examples are from taking a genetic variant, taking it through expression maps to function, often making in vivo models to then human cells, and that process has roughly taken about 3 years. What I'd like to propose is that now that we have better knowledge of better applications to handle human cells, and in the next few examples that should appear in press soon, we've been able to take genetic variants directly to human cells, identify function, build models, and then use small molecules to either correct the phenotype or inhibit the phenotype. And that process should take about 6 months. Roughly, the importance of this is that for non-coding variants, my view is that it's a waste of time doing mouse experiments because you have to do this in human cells. So I think I'll end there. Sorry.
Since you talk about pathways, I think, so one way to think about the whole problem we're discussing today is, and tomorrow is, what are we missing to speed up the process, or what are the things that we need to drop, and what are the things we need to include? And that you said, for testing regulatory variant mouse, it might not be relevant. Another question is how correct or relevant are the current resources that we have? So for example, assignment of pathways, the grouping of genes into whatever categories we have. Is it relevant? Is it accurate? What's the variance in that accuracy from pathway to pathway? And are they useful as they are, or do we need to generate data to change them so that they become relevant for whatever we're thinking in the future?
So we need to generate data to change them because the current pathway maps are not very useful. The current pathway maps are based on 2,000 commonly worked on genes that are published in PubMed. 18,000 other genes have not been mapped to pathways, and so one needs to revisit this. So the example that I tried to highlight was with autophagy, where if you look at the PubMed as a data source, 2 or 3 genes have been implicated in autophagy. But when we did a screen, we place 18 new genes in the process of autophagy in a relevant cell type that contributes to disease.
Ewan, just on that second, that point you made there, actually in Reactome, it's over half of the genes are present in a pathway. So half of human genes, about 11,000. But I had a slightly different question about to what extent that lovely network diagram you had was compelling or really interesting to see. Do you think, how do you like to think about environmental preservation in this network, especially in a disease which clearly has this interplay of gut microbiome and other things? How do you think about placing environmental components in with the genetics?
Yeah, so that's important. And so I didn't present slides here, but we've obviously, with all the genetic variants, we've taken the microbiome as well as diet, which is an important component in gut diseases, into account. The example was in ATG16L1, T300A. Now we've identified a specific microbiome that now when you combine genetics with the microbiome, you get disease in a mouse. And so clearly, and in the slide that I showed you about single cell mapping of cell types and genetic variants, we're also collecting microbiome in situ from the 2 different sites, from the inflamed and non-inflamed, to see whether we can make these associations better.
Last one.
That was a really lovely talk. Maybe just from a philosophical level, if I was to grab 10 random people from the street, not in a city like Oxford, but in a normal city, and ask them, what is your expectation from genetics? I hope most of them would say new medicine. And I was really pleased that you introduced the concept of chemical biology as kind of that key translator from genetic insights to drug discovery. And the example you gave at the beginning, the kinase example, was really neat. Could you comment just a little bit on what is the reason people have acted upon that particular discovery from a therapeutic perspective versus all the other targets that could have come from the IBD genetics world? And how can we accelerate that systematic chemical translation? Because I feel, and I'm biased working in drug discovery, that's often the bottleneck that we face.
Yeah, so with the IL-10 pathway, the way we sort of convinced pharma was that while IL-10 and IL-10 receptor have been genes associated with disease risk, in common disease as well as in sort of children with early onset disease. We were able to identify that there were 4 other genes within the IL-10 signaling pathway that were also associated with disease risk. And in fact, when we did the screen, we looked for molecules that increase IL-10 and decrease IL-12, but as a readout, we used the other 2 genes as markers within the primary cell. So we knew we were impacting a pathway that went from IL-10 receptor all the way to transcriptional and central regulation of the pathway. And that was a lot easier sell to pharma. Obviously, with pharma, the challenge is, genetics affects only a small number of people. How do you sort of make drugs to impact the broader disease phenotype? And my response to them has been, yes, disease affects only, genetics is focused on only a small number of people, but that pathway is important for all of inflammatory bowel disease. And you can sort of obviously see where I'm going. If I have many genes in the pathway, even for the person the pharma scientist who doesn't want to believe in me is forced to believe because I've told him that now there are 5 genes.
And you've got a couple of other.
Yeah, so we've been able to sort of move something like 4 other pathway maps and convince large pharma to take the bait. And this is all only for IBD. We've sort of now started expanding into other autoimmune diseases, and because I'm at the Broad, I'm forced to start thinking about schizophrenia, and I think we'll get there.